Hi, my name is Megan Yang, and my project is inhibiting the TRPM7 and ERK pathways in gastric and bladder cancers. So, gastric cancer occurs when the cells along the lining of the stomach start to proliferate uncontrollably, beginning with the innermost layer and spreading outwards as it develops. It is currently the second most common cancer worldwide and accounts for between 3 and 10% of all cancer-related deaths. Bladder cancer occurs similarly, starting with the innermost layer and spreading outwards as it develops, and it's currently the fourth most common cancer in men. So my first pathway was the transient receptor potential cation channel subfamily M member 7, or TRPM7. It is a chanzyme, meaning it is both an ion channel as well as an enzyme or kinase. It has a dominant role as a magnesium and calcium transporter. So, it has also been shown to contribute to the development of several cancers, such as breast, pancreatic, and glioblastoma cancers. Recent studies have also suggested a relationship between TRPM7 and cell tumor proliferation, differentiation, migration, and invasion. The TRPM7 could potentially have a major role in gastric cancer, as magnesium levels have been shown to be elevated in cancer stomach tissue in comparison to healthy stomach tissue. Moreover, because of its role as a magnesium transporter, TRPM7 is necessary for the magnesium-dependent survival of gastric cancer cells. It is also involved in the ginsenoside-induced apoptosis. Furthermore, a study had demonstrated that human gastric adenocarcinoma cells have TRPM7 channels, which influences their cell survival and proliferation. In cancerous bladder tissue, the TRPM7 has been shown to be overexpressed in comparison to the non-cancerous neighboring tissues. Furthermore, as the TRPM7 levels elevate, so do the metastasis and prognosis levels. So my second pathway is the mitogen-activated protein kinase, or MAPK. MAPK is a serine specific protein kinase that has the ability to influence things like proliferation, apoptosis, cell survival, and usually responds to extracellular signals. So when it's abnormally regulated, it oftentimes becomes involved in the occurrence and progression of cancer. So within MAPK, there are three subcategories, which are the extracellular regulated kinases, or ERKs, the C-Jun terminal kinases, or JNKs, and the P38 MAPKs, and amongst these three, the ERKs have been shown to be involved in carcinogenesis, while the other two are just involved in cellular signaling. Mutated ERK pathways actually exist in more than half of human cancers, suggesting a relationship between the ERK pathway and tumorigenesis. The ERK pathway also influences metastasis due to the fact that metastasis requires cell motility, which is dependent on cellular adhesions, and this pathway regulates the epidermal growth factor receptor, which has the ability to induce the disassembly of focal adhesions, causing metastasis. So the link between these two pathways are that TRPM7 can regulate metastasis via the MAPK pathway, and it also, when the TRPM7 is silenced, the metastasis levels and phosphorylation levels of the MAPK pathway are also lowered. Additionally, the TRPM7 calcium influx is necessary for ERK activity. So in my project, I used 4 treatments, 3 of which are derived from plants because most chemotherapeutic drugs are cytotoxic to not only cancer cells but to normal cells as well. Therefore, treatments involving herbs are becoming a novel and more popular treatment for cancers. So my first treatment is ginseng, which is a popular and well-known herbal remedy believed to restore one's well-being, and has these things called ginsenosides, and a study had suggested that ginsenoside-mediated cellular injury is affected by TRPM7 channels. More specifically, the ginsenoside RD has shown to be able to inhibit the TRPM7 ion channels in kidney cells, and as well as lower phosphorylation levels in the MAPK pathway. So my Second treatment was the NS8593, which acts as a negative gate modulator for the TRPM7 pathway. It is also very specific to this pathway since it is ineffective in inhibiting homologous ion channels such as TRPM2, TRPM4, and TRPM6. A study had also shown that this substance had interfered with the magnesium influx and thereby decreasing cell motility in cultured cells. This will aid in determining whether or not TRPM7 is an effective target for treating gastric and bladder cancer cells. So my third treatment is curcumin, which had been shown to be able to inhibit proliferation, induce apoptosis, and have other anti-tumor effects on various cancers such as lung, breast, and colon cancers. And in colon cancer cells, has been shown to be able to activate the P38 MAPKs and JNKs without activating the ERKs, as well as suppress anti-apoptotic proteins like BCL1, BCLXL, and MCL1. My fourth and final treatment is Icarin, which is a flavonoid extracted from a plant called epimedium and is able to indirectly inhibit the ERKs. So the way it does that is it blocks the signaling for the RAF1 kinases, which typically activates the MEKs, which in turn activates the ERKs. So I conducted 4 assays for my gastric cancer cells. I did a TRPM7 and ERK ELISAs since studies have shown that there is a correlation between the protein levels and the activity levels of TRPM7 and ERK, as well as a cell proliferation assay and an apoptosis assay. And for bladder cancer cells, I only used the TRPM7 ELISA since ERK doesn't seem to be very significant in the progression and development of bladder cancer. And I also did the cell proliferation and apoptosis assays. So my 4 treatments were tested in combination as well as individually and compared to a negative control, which was no treatment at all, and a positive control, which was docetaxel, which is a common chemotherapeutic drug currently used a lot. And my conclusion stated that for gastric cancer, the most effective overall treatment was the combination of the 4 treatments, including docetaxel, which was statistically significant when compared to, and the most effective individual treatment was NS8593, which was also statistically significant compared to the docetaxel. And for the bladder cancer, NS8593 was actually the most effective overall treatment, and that was also statistically significant to the docetaxel. So with further research, these results could provide to develop a more effective and less costly treatment than current chemotherapeutic drugs, since those drugs can currently cost up to $65,000 in a single month. And moreover, these results can help develop treatments for other cancers that have and are involved with the TRPM7 ion channel, such as pancreatic, breast, prostate, glioblastoma, and ovarian cancers, and the ERK pathway, which is overexpressed in cancers like prostate and gallbladder cancer. So future research would include things like a migration assay to test the effectiveness of these treatments on inhibiting metastasis, as well as starting to test on animals and using animal models to see if there are any fatal consequences of these treatments in vivo. Thank you for listening to my presentation.